Namaskar, my dear students. Today we will be starting with the second part of the chapter photosynthesis. In the very first lecture, I have told you that photosynthesis is the phenomena which can be studied under two headings, that is the machinery of the photosynthesis and the mechanism of photosynthesis. Before that, we discussed the definition of photosynthesis, a brief history, how this process has evolved to the present date knowledge of photosynthesis, and we also discussed about the generalized reaction of the process of photosynthesis. In addition to that, I talked to you about the machinery of the photosynthesis, in which we discussed the various ingredients which are required for this process to take place, that is chlorophyll, sunlight, water and carbon dioxide. All these things we discussed in detail, their sources and where they are present and what role they play. Today we are going to start with the second phase, that is the mechanism of photosynthesis, in which I have to tell you that the total process of photosynthesis, it includes two phases and three main goals, that is the light reaction, which is called as the light capturing reaction, obtaining the molecules of the energy which are to be utilized in the dark reaction. The term dark reaction is more precisely called as the light independent reaction, since this is the reaction that does not take place in the light, but in fact it does not require the light, during which the energy currency obtained during the light reaction is used for the synthesis of the organic compounds from the atmospheric carbon. Light reaction or the photosynthesis, photochemical phase. Now, let us discuss where does they actually take place. In fact, as already discussed in the first lecture, that chloroplast has two areas. One is called as the grana, which consists of the thylakoids. Grana are the sites of the light reaction. Grana contains the light trapping pigment, the chlorophyll, and stroma is the gel-like matrix of the chloroplast. Stroma contains enzymes which are needed for the dark reaction or the light independent reaction of the photosynthesis to take place, and it is this place, the stroma, where the dark reaction takes place. Now let us start with the light reaction. As you can see, this is the diagram of the light harvesting complex. The light reaction or the photochemical phase includes light absorption, water splitting, oxygen release and formation of high energy chemical intermediates, ATP, adenosine triphosphate and NADPH, nicotinamide adenosine phosphate hydrogenase. Several complexes are involved in this process. The pigments are organized into discrete photochemical light harvesting complexes, LHC, light harvesting complex, within the photosystem 1 and photosystem 2. Photosystem 1 and photosystem 2 I have already discussed with you, but again I will be making a brief note over here. These are named in the sequence of their discovery, that is photosystem 1 discovered first, and not in the sequence in which they function during the light reaction. Light harvesting complex is made up of hundreds of pigment molecules bound to the proteins. Each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system, which are also called as the antennae. As you can see in the figure, these are these pigments help to make photosynthesis more efficient by absorbing different wavelengths of the light. The single chlorophyll A molecule, which works as a reaction center, This reaction center is not a different molecule, but this is different in both the photosystems. It is just like the other molecules, but any molecule can function as the reaction center. Photosystem 1, the reaction center chlorophyll A has an absorption peak at 700 nanometers, hence that is called as the P700. I repeat once again that in photosystem 1, the reaction center chlorophyll A molecule has an absorption peak at 700 nanometer. Only that is the difference. While in photosystem 2, it has absorption maximum at 680 nanometer, hence called as the P680 also. In photosystem 1, in here you can see the difference between the PS1 and the PS2. The thylakoid membrane of the plant chlorophyll have two different kinds of photosystem, photosystem 1 and photosystem 2. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into orbit farther from the atomic nucleus. These electrons are picked up by the electron acceptor, which passes them to the electron transport system consisting of cytochromes, and as you can see in this figure and in detail you can see here. Now the movement of the electron is downhill in terms of an oxidation reduction or redox potential scales. What I mean to say, the electrons are not used up as they pass through the electron transport chain, but they are passed on to the pigments of the photosystem 1. Simultaneously, electrons in the reaction center of photosystem 1, they are also excited when they receive red light of wavelength 700 nanometer, because it is PS1, and are transferred to another receptor molecule which has a greater redox potential. These electrons are then moved downhill because it keeps on losing its energy and keeps on moving downhill. This time to molecule of energy rich NADP. In addition of these electrons reduces NADP to NADPH plus H positive. This whole scheme of transfer of electron starting from PS2 uphill to the acceptor down the electron transport chain of PS1, excitation of electrons, transfer of another receptor, and finally downhill to NADP, causing it to form NADPH, is called as the Z scheme. As you can see in the figure, this is looking like a Z. Due to its characteristic shape, it is called as the Z scheme of photosynthesis. This shape is formed when all the carriers are placed in sequence on the redox potential scale, that is according to their reducing or increasing energy. If you place them, it forms a Z scheme of photosynthesis. Now as you can see, there occurs the first of all, the prerequisite is the splitting of the water. Now you can ask a simple question, how does PS2 supply electrons continuously? The electrons that are moved from photosystem 2, they must be replaced. This is achieved by the electrons available due to splitting of water. The splitting of water is associated with PS2, photosystem 2. Water is split into H positive ions, O oxygen and electrons. Three things: H positive, O oxygen and electrons. This creates oxygen, which is one of the net products of the photosynthesis. As you remember, in the generalized reaction of the photosynthesis, oxygen was one of the end product, and here is that oxygen that is produced. The electrons needed to replace those removed by photosynthesis system 1 are provided by photosystem 2. Here we need to emphasize that the water splitting complex is associated with PS2, which itself is physically located on the inner side of the membrane of thylakoids. Then where are the protons and oxygen formed likely to be released? In the lumen or on the outside of the membrane? You just think it before I continue with the other phase. And this is the cyclic photophosphorylation, which involves only photosystem 1. And here what happens, that only ATP is produced, and this is cyclic. It means the electron is returned back, and therefore continuous supply of the electron by splitting of water is not needed. To understand it in a better way, let us see what are the differences between cyclic photophosphorylation and non-cyclic photophosphorylation. In cyclic photophosphorylation, only photosystem 1 is involved, whereas in non-cyclic, both the photosystem PS1 and PS2 are involved. Regular supply of the water is not required in cyclic photophosphorylation, because once the splitting occurs and electron becomes the continuous source, whereas the photolysis of water is required in non-cyclic photophosphorylation. Since no photolysis of water is occurring continuously in the cyclic photophosphorylation, hence oxygen is not involved in cyclic photophosphorylation. However, it is evolved in non-cyclic photophosphorylation. In cyclic photophosphorylation, since the second step does not take place, hence NADPH is not synthesized, only ATP. Whereas in non-cyclic, NADPH is synthesized. Cyclic photophosphorylation used to produce additional ATP in order to meet cell energy demands, whereas products in non-cyclic photophosphorylation can be used for light independent reaction. Now, after cyclic and non-cyclic photophosphorylation, let us conclude what happens at the end of the light reaction. First, ATP is generated, which is called the energy currency for the next process to take place. NADP is reduced to NADPH, hence this is called as the reducing power because hydrogen is added. And both ATP and NADPH together are called as the assimilatory powers, because both of these are required for assimilation of organic compounds from inorganic atmospheric carbon in the dark phase of photosynthesis. Both of these are used. ATP is used in phosphorylation of 3 molecules of phosphoglyceraldehyde into 1,3-biphosphate glyceraldehyde. NADPH reduces 1,3-biphosphate phosphoglyceraldehyde into phosphoglyceraldehyde, and oxygen is evolved. This is the net result of the light reaction. Now, after the light reaction has taken place, there takes place the dark reactions. The dark reaction, also called as the Kelvin cycle, because the things which are used in the light reaction are dark reaction are re-synthesized back. The dark reaction uses ATP from the light independent reaction and carbon dioxide to produce sugars. This is also called as light independent because no light is needed. Now, a very simplified way, in a simplified way, it is represented that ATP is used and gets converted to ADP, and the energy so released is used for converting carbon dioxide of the atmosphere into sugars, and the reactants which are used, they are returned back by the Calvin cycle. We learnt in the light reaction that ATP, NADPH and oxygen uh, are the products of the light reaction. Of these, oxygen diffuses out of the chloroplast, while ATP and NADPH are used to drive the process of synthesis of the food. More accurately, formation of sugar. This is called as the biosynthetic phase of the photosynthesis. This process does not directly depend on the presence of the light, but is dependent on the products of the light reaction. Let us see how ATP and NADPH are used in the biosynthetic phase. We saw earlier that carbon dioxide is combined with water to produce sugars. It was of interest to the scientists to find out how this reaction is proceeded, or rather what was the first product formed from the carbon dioxide. When carbon dioxide is taken into the reaction just after World War II, among several efforts to put radioisotopes to beneficial use, the work of Melvin Calvin, that is exemplary work. The use of radioactive carbon, that is carbon 14, he used in the algal photosynthesis studies and led to the discovery that the carbon dioxide fixation product was 3-carbon organic compound. He also contributed to the working, but the complete biosynthetic phase, when it was discovered, it was called as the Calvin cycle after his name. The first product identified was 3-phosphoglyceric acid, or in short 3-PGA. Hence, it is known as the C3 pathway. Scientists also tried to know whether all the plants have PGA, phosphoglyceric acid, or as the first product of carbon dioxide fixation, or whether any other product was formed in other plants. They conducted a number of experiments, and since carbon dioxide assimilation during photosynthesis was said to be of two main types, these plants in which the first product of the carbon dioxide was a 3-carbon compound, that was called as the C3 pathway, and those plants in which the first acceptor was oxaloacetic acid, or the 4-carbon compound, were called as the C4 pathway. Now let me mention here that the studies were unexpectedly done, and they showed that the acceptor molecule in the 5-carbon ketose sugar ribulose biphosphate, it was a 5-carbon compound. And what happened, that they believed that since the first product was C3 acid, the primary acceptor would be 2-carbon compound, and ultimately ribulose biphosphate was identified as the first compound. Now, the ribulose biphosphate carboxylase is the enzyme that is involved in this reaction, and this is the most abundant enzyme and is returned back or regenerated back during the Calvin cycle. Calvin and his co-workers studied about the whole pathway and showed that the pathway operated in a cyclic manner. The ribulose biphosphate was regenerated. Now let us see how in this cycle, as it is mentioned in the figure, that how does the pathway occurs. Now, for ease of understanding, Calvin cycle can be described under 3 stages: carboxylation, reduction and regeneration. First of all, carboxylation. It is the fixation of carbon dioxide into stable organic intermediate. Carboxylation is the most crucial step of the Calvin cycle. where carbon dioxide is utilized for the carboxylation of ribulose biphosphate. This reaction, it is catalyzed by the enzyme RUBP, ribulose biphosphate carboxylase, which results in the formation of two molecules of 3-phosphoglyceric acid. Since this enzyme has an oxygenation activity, it would be more correct to call ribulose biphosphate carboxylase oxygenase or ribusco. This is most abundant enzyme. The second phase is the reduction. What happens in this stage? There are the series of reaction that leads to the formation of glucose. The step involves utilization of two molecules of ATP for phosphorylation and two NADPH for reduction per carbon dioxide molecule fixed. The fixation of 6 molecules of carbon dioxide occurs, and hence 6 turns of cycle are required for the removal of 1 molecule of glucose from the pathway. The third step would be dealt under the heading regeneration. A regeneration of carbon dioxide acceptor molecule is crucial if the cycle is to continue uninterrupted. The regeneration step requires 1 ATP for phosphorylation to form ribulose phosphate back. Hence, for every carbon dioxide molecules entering the Calvin cycle, 3 molecules of ATP and 2 molecules of NADPH are required. It is probably to meet this difference in the number of ATP and NADP used in the dark reaction that the cyclic phosphorylation takes place. Now, let me tell you that balance sheet of photosynthesis. Balance sheet means what is taken and what is returned back. That is how much ATP and NADPH are required to make one molecule of PGA from carbon dioxide. Each turn, because 6 cycles are there, 6 turns are there, and in each turn, one carbon dioxide molecule is fixed. Phosphoglyceraldehyde is a 3-carbon compound, takes 3 turns to make each molecule. Each turn of the cycle, that 3 ATPs, out of which 2 in step 1, because I discuss you with the 3 steps, that is carboxylation, reduction and regeneration. They are required 2 in step 2 and 1 in step 3 and 2 NADPH in step 3. So in total 6 carbon dioxide molecules are added, 18 ATP are added and 12 molecules of NADPH are added. And as a result, the outcome is 1 molecule of glucose, 18 ADP converted from ATP, and 12 molecules of NADP are returned back. This is the balance sheet of the photosynthesis of the C3 pathway. Next time, when I will continue with this topic, we will discuss the C4 pathway, that is another alternative pathway of photosynthesis, and we shall discuss the photorespiration and factors affecting the process of photosynthesis. So that's all for today. Till we meet next time, till then, thank you and stay safe, my dear students. This is the process of photosynthesis is definitely, it requires a concentrated mind to understand the process, and a lot of calculative mind and imaginatory power as to how does it occur, and is very important. So I would suggest you that you go through the topic twice in order to make a perfect understanding, and still if there is any query you can ask me on my phone number or leave your message or the query at my email address. Thank you. Stay safe. Stay